This method can help answer key questions regarding the organization of astrocytic networks in brain structures.
The main advantage of this technique is that it proposes an unbiased method to count labeled cells and document their anatomical organization within a defined structure.
Open ImageJ select the desired file, and click OK in the Bioformats Import Options window. To redefine a Z-stack that will contain only the optical slices needed for the final Z-stack, first select STK and Z project, and then click on the Stack knob in the toolbar. Select Max Intensity in the projection type setting. Save the file and name it Stack File. If the imaging file contains several channels, use Image Color and Split Channels to show only the channel with the image of the astrocytic network. Check the pixel settings of the image by selecting Image, Properties, Pixel with 1 4-pixel dimension. Then use the Subtract Background tool by opening Process and Subtract Background to remove background biosite and labeling. Use the Preview function to set the rolling ball radius, which is generally set at 50 pixels. If further noise reduction is required following the Subtract Background step, select Process, Noise, and remove outliers. Select Bright in the Which Outliers setting, and use the Preview function to set the radius and threshold.
Be careful.